Hi everyone, welcome to Medical Microbiology. So today we will continue chapter 12, uh, chemotherapeutic drugs. And today we are in lecture number 2, which is we will talk a little bit about the drugs and their targets. So remember this picture, this diagram. Again, if we talk about the bacterial cell, it has cell membranes, uh, DNA, which is chromosome, of course, ribosome for protein synthesis, and some metabolic pathway inside the bacterial cell. So this is we're talking about process or structure of bacterial cell, which is our target for taking the drugs or the antibiotics. So there are some drugs that will inhibit the cell wall. There are some drugs that will inhibit the cell membranes. There are some drugs that used for blocking DNA or RNA replication. There are some drugs that's used for blocking the ribosomes. And then there are some drugs that's used for blocking the metabolic pathway inside the bacterial cell. So if this structure and also process being blocked, then the bacterial cell eventually will die. Okay, so now we start with the one that block the cell wall. Okay, bacterial cell wall. So remember, bacterial cell wall made of peptidoglycan. So this become the target for the drug, which is destroying the peptidoglycan. Examples of that drug is in the class of penicillins, and mostly this one will be very effective for gram-positive bacteria. Okay, remember gram-positive bacteria, they have thicker peptidoglycan. Therefore, this peptidoglycan will be easier to be destroyed by the penicillin. Compared to the gram-negative bacteria, they have outer membranes that kind of protect the peptidoglycan from the drugs. So usually the drugs that effective against the cell wall mostly used for gram-positive bacteria. Like penicillin, for example, is used for Staphylococcus, Streptococcus, which is gram-positive bacteria. And they usually not used for like E. coli infection because E. coli is gram-negative bacteria. But there are also some broad spectrum of penicillin that can be used for both gram-positive and gram-negative bacteria. But just remember, penicillin mostly more effective for gram-positive bacteria. So this is really important when you do the experiment identifying whether the bacteria is gram-positive or gram-negative. It is also used for giving the correct drugs for treatment of this infection. So for example, the doctor will not give prescription for gram-positive bacteria infection if the infection is actually caused by the gram-negative bacteria. Okay, this is what happens when the cell walls are broken. So like penicillin will break the cell wall. Then what happens? The water will enter inside the bacterial cell, and bacterial cell is become bigger, and finally, it will explode. This is from the electron microscope. So what happened with this bacteria that the cell walls has been broken, it's become bigger because of water is actually enter into their cell, and some of them is already explode. So they die because their cell become explode due to the entrance of water into their broken cell. Now the one that destroy the cell membranes, the example is polymyxin. So this polymyxin is actually separate the lipids, the phospholipid bilayer, and causing also leakage to the cell. Okay, so this one usually used for gram-negative bacteria. So this polymyxin is actually break this cell membrane. The drug that inhibits nucleic acid synthesis, example is chloroquinone and quinones. Some of them, for example, chloroquinone is actually bind to the double helix DNA, so the DNA will not be functional. And quinone, for example, they actually inhibit the helicases. Remember, when the DNA start to replicate or duplicate, they need to be unwind, because this is double helix structure. Okay, so in order to be replicate or duplicate, this double helix has to be open. So like it's to be open like that. So therefore, the new DNA will be made over here. Okay. Now, in order to open this double helix, they need enzyme is called the helicases, and quinone is actually block this enzyme. Therefore, the bacteria DNA will not be able to replicate. The drug that block the protein synthesis, okay, several of them, like for example, aminoglycoside will block the 3S subunit, which is smaller subunit. So if you remember this is bigger subunit, and this is smaller subunit. So this is 50S, and this is the 30S unit. So aminoglycoside is actually block this smaller subunit. So tetracycline, for example, will block the larger subunit. Okay, so this is example. This is aminoglycoside. Is actually here. Okay, so they attach to the smaller subunit, causing the messenger RNA will not be able to convert it or translate it into the protein. So there is no protein production over here. For chloramphenicol, for example, this is on the blocking the larger subunit. So when it is blocked, then messenger RNA will not be able to translate it into the protein. Other like chloramphenicol, tetracycline, erythromycins, they are blocking between this bigger and smaller subunits. So the process will be blocked over here. So no protein production. And the last one is drug that affect the metabolic pathway. Example is sulfa drugs and rifampin. So sometimes they do this process by competitions with the process, with the substrate, for example. Okay, this is examples. The use of sulfa drugs and trimethorphan. So sulfa drug usually used for the first process over here, which is competition with the substrate. Okay, so this is the normal process for bacteria to produce the product over here. So this product is amino acid that is important for them. So they need this amino acid in order to perform their process, maybe growing or reproduction. So the normal process is they have the substrate called the PABA or PABA, and this PABA will enter or will combine with the enzyme, okay, and then they will produce the intermediate products over here. And then after that, the intermediate products will be used to make these final products. Okay, so this is the normal process. So when they're able to produce this, then they will growing very fast, they reproduce, and etc. Okay, so the drugs, sulfa drug, for example, is used to block this process over here, compete with the substrate. So this is substrate is look like, okay. All right, so this is the normal process. The substrate will bind to the enzyme. So this is enzyme, and finally it will produce the intermediate product, okay, which is this one. Okay, so the process then continue to produce the final products over here, the amino acid. Now, when the sulfur drug coming, so this is sulfur drug. If you see the sulfur drug is very similar with the substrate over here. It's kind of similar, right? So that's the reason why, you know, this sulfur drug actually compete with the substrate to bind to the enzyme, right? So when the sulfur drug that actually bind to the enzyme, then there will be no intermediate product of here. So naturally, you know, normally, you know, the substrate will enter into the enzyme and produce this. However, because the sulfur drug enter into this combination, okay, so enzyme and sulfur drugs complex, then there will be no production of intermediate product. So if there is no intermediate product, then there will be no final product. So that's the sulfur drug. The trimotrophin, okay, is work on the second process over here. So let's say there is no sulfur drug, so the bacteria able to produce the intermediate products over here. Then what happens? The trimotrophin enter into this metabolic pathway and block this process. So if the process is blocked, then there will be no amino acid production. So this is how sulfur drugs and trimotrophin block the metabolic pathway. They're actually blocking different side, not the same side of pathway. Yeah, I think that's enough for lecture number 2. We will have a next lecture, especially we're gonna talk about the antibiotics, which is the drugs that used for bacterial infection. Okay, I see you later. Goodbye now.